right, let's go ahead and get started. Thank you for joining us today. My name is Kathy Gredling. I'm Executive Associate Dean for Faculty Affairs at the School of Medicine at Emory University. But I also lead the collaboration of multidisciplinary team science part of the CTSA. And so as part of our efforts to help enhance collaboration across our 4 institutions and otherwise in Atlanta, we hold these blue sky groups, and blue sky groups are they're unstructured meetings. They work best when people participate and talk to each other. We'll ask an expert on the topic. In this case, it's Tamina Anderson, who's a leading researcher in organoid development, to just sort of set the stage for us about what we're talking about. And then we'll ask each of you to share your research. And when you share your research, I'd like you to tell us what you're working on and what you're looking for in a collaborator. We'll encourage you to use the chat to follow up with people who say something interesting to you. And hopefully by the end of the time, you'll have identified a new collaborator. We'll also, if we have time at the end, have sort of a general discussion about what the burning issues are in this area, known funding opportunities, especially given the changing priorities of the NIH, and where we might see some cross-institutional collaborations. So hopefully you'll enjoy the next hour and a half. You'll be able to learn from each other and to share your own experience. Next slide, please. We have a pilot grant program too to help enhance your collaboration. So if you meet someone today and you decide that you're going to apply for external support, we have a very small grant to help you maybe have those meetings, those first meetings when you're trying to plan the grant or invite someone to speak. But the idea is that these would seed an external application. So if you're interested in applying, we'll show you the QR code again at the end. And so right now, I'd like to turn it over to Tamina Anderson. She is a researcher at Emory University. She studies spinal cord and motor system, and especially how cell-to-cell interactions contribute to disease. So she uses stem cell-derived organoids and assembloids to answer these questions. And she's going to set the stage for us. So, Tamina.
Venna, you are on mute.
Tamina, you're still muted.
There
we go.
Hi, I'm really sorry. My computer froze, of course. And now
We can hear you now and we can see your slides.
okay. I'm so sorry. Okay, I'm so sorry. Let's start again. Okay. You can now.
All good.
And you can hear me.
All good. Yep.
Okay. Thank you. Thank you so much for the invitation. Apologies for the minor glitch there. So yeah, I'm excited to be here today and tell you a little bit about organoids. So yeah, I'm an assistant professor at Emory University in the Department of Human Genetics. And my lab uses mostly brain spinal cord organoids to study development and degeneration of the spinal cord. And previous to my time here at Emory, I was a postdoc at Stanford University in Sergio Pascal's lab, where I developed a lot of the current neural organoid models, spinal cord, characterized a lot of the cortical organoids and benchmark them, compare them to primary tissue, and also develop some of the assembly models. And so for today, I wanted to just kind of put everyone on the same page on what organoids are, what can they be used for, and really think about some of these considerations about organoids. We have all seen the exciting, the Nature, Science, and Cell papers showing all the great things that organoids can do. But organoids also are pretty new. And they still require a lot of considerations and a lot of things that maybe we don't have to think about with other model systems. So let's start. What are organoids? So organoids are complex 3D in vitro derived structures that mimic key functional, structural, and biological features of an in vivo organ. And they develop through a self-organization process. And self-organization can be defined as the capacity of a cellular system that initially lacks an order structure to spatially rearrange under the guidance of system autonomous mechanisms. And I like to emphasize this self-organization because I think it is quite an important point of organoids compared to other cellular systems like in 2D. And there are a ton of different organoids out there. Every month there's a new type of organoid that is being published. Again, my expertise is brain, but and most of my slides are probably going to be about brain, but I know that a lot of you out there today are doing other things that are not brain. So, I wanted to put this out there so that that is something that we think about. So again, why are we going to use organoids? So they're cool, but there's other model systems. There's more simple model systems that we can use. We can use 2D cultures. So, okay, we use organoids because, yes, we can limit the need for animal experimentation. Gives us access to human cells. We can use patient-derived cells, and so we can study disease that is specific to a patient. Gives us accessibility to experiments, so we can do quite a lot of things with organoids. Importantly, it gives us cellular diversity. Again, in a cortical brain organoid, we can get neurons and astrocytes and different types of neurons. So we can look at multiple cell types that interact. They have a cytoarchitecture that sort of resembles the cytoarchitecture of the organ that they're trying to mimic. Again, because we have this cell diversity, it means that we can study cell-cell interactions. And we have a complex extracellular environment that, again, we cannot have in 2D. And one of the things that sometimes we forget about, but we can keep these things in culture for quite a long time. And that allows us to study things that maybe we can't do in 2D. We can study cells that are more mature. And we can study cells that are generated later during development. And in addition to being able to study cell-cell interactions, we can also start to study regional interactions and connectivity between regions. So by assembling two different region-specific organoids together, it allows us to study interactions between these two different regions. And so we can generate them separately and then assemble them together. Okay, how do we make organoids? So the successful derivation of organoids depends on three main characteristics: cell type of origin. So we can start with pluripotent stem cells, so ES or iPS cells, or we can start with tissue-derived adult stem cells. So most of the work that I do is with iPSCs, and most of it is derived from patient cells to study disease. Physical characteristics of the culture environment. So this, means where do you put them? Are they in suspension? Do they have ECMs to help them grow? Are they in a bioreactor? and then the addition of exogenous signals. And so we used the knowledge that we have basically of years of developmental biology, where we understand the factors that are important to pattern specific regions of interest, and we add these in our cultures to mimic that development. And so this is an example of starting with iPSCs, we can push our iPSCs into all three germ layers: endoderm, mesoderm, neuroectoderm, using our exogenous factors to give rise to a bunch of different types of organoids that can be matured in different ways, with matrices in suspension, being spun around. And the important thing to note is that there isn't one best thing to do this. There are tons of protocols out there, and different people for different protocols and different questions will use different methods. Another thing that is very important in the field, now we're not talking about I have a mouse and I have a brain, I know exactly where my hippocampus is. Now I need to make this organoid, and I need to be able to show that the organoid has what I need to ask the question that I want, right? So I want to ask, do they have the expected cell types? And the way that we normally do this is single-cell RNA-seq, qPCR, immunos. We also want to ask, do they have the expected cytoarchitecture? And this will depend and vary depending on the type of organoid that you're working with. So in a brain organoid, you might look for these ventricular zones that have the progenitors in them, and then surrounding them have the deep layer and superficial layer neurons. But in other types of organoids, you might be looking for budding or other structures. Importantly, do the cells perform the functions that you expect? Again, we are trying to mimic a cell that performs a function in an organ. And if we want to be able to model these cells, we want to know that they're performing this function. And so, for example, this is an example of a spinal cord organoid that I generated when I was a postdoc. And if we make spinal cord organoids that have motor neurons, I want to be able to show that the motor neurons can innervate a muscle and make it contract. And so we've shown that in many different ways, but this is assembling to a mouse limb bud. And lastly, do the cells resemble their in vivo counterparts? Again, we want to show that they are similar to a human in vivo cell type to be able to say things about human biology. So what are some promising organoid applications? Again, different things for different types of organoids. So for example, if we start with adult stem cells, and that's mostly non-brain, but we can, again, study patient-specific pathology, host microbe interactions, tissue growth and homeostasis, and drug screening. I think this one is quite a big one. So there's a big success story with gut organoids that we can get adult stem cells and specific from patients. And starting with ES cells or iPS cells, similar things, but again, we can think about more developmental biology questions that maybe we don't think about with the adult stem cell organoids. But similarly, we can study patient pathology, disease model, host microbe interactions. We can transplant them. And people use transplantation as a way of maturing the organoids a little bit more, also getting them vascularized, because that's one of the limitations of the organoid systems, is that currently they're not vascularized. So talking about limitations, again, organoids are great, and they can give us a lot of answers about human biology, human-specific biology, disease-specific biology, but there are a lot of limitations that we need to think about. One of the biggest ones that you've probably all heard about is heterogeneity and reproducibility. And this is very important to think about when you're starting an experiment. So how am I going to limit this variability that I'm going to get? And the variability can come from organoid to organoid, it can come from batch to batch, and it can come from iPSC lines. So, and quite a big portion of our variability is indeed coming from our different genetic backgrounds. So when we use different IPS lines, we are basically using cells from different people that have different genetic backgrounds. And this is something that we need to think about. And what are the best controls that we're going to use for our experiments? You know, if we are doing disease modeling, are we going to use patient cells? Are we going to use a control cell line that we're going to introduce a mutation in? How many controls, how many healthy controls, how many patient derived cells do I need? And these are things that are very important to think about from the very beginning of the experiment so that you actually can get something out of it. Another limitation, limited cell composition and organization. You know, we lack a lot of cell types in the organoids. And sometimes I tend to think about as an advantage in that it gives you an ability to add them later. And so you can study, you know, for example, we study ALS in the lab, and microglia is an important aspect of ALS. And we don't have microglia in our spinal cord organoids, but I like to think of that as an advantage because we are able to study cell type specific processes that are independent of microglia and inflammation. And we can then add microglia if we want to study that aspect of the disease, but we don't have to. And you know, another big problem is this limited knowledge of developmental pathways. So especially when starting with iPSCs, we need to introduce the factors that will generate the cell type of interest. But if we don't know what those factors are, then you might be stuck and not be able to generate the cell type that you want to study. And limited maturity and function. And this is something that, again, especially for iPSC-derived organoids, we are starting from the beginning, and this are developing, and maturity takes many, many years in a human. And there are a ton of approaches that people are working on to kind of overcome these limitations, you know, including vascular networks, using bioreactors, using different 3D matrices, co-culturing, or using organ-on-chips that are a little bit more structured compared to organoids. And, you know, there's still a lot to do. And I think when I think about limitations in the organoids, really, I think about opportunities, and that there's still so much that we can do in the field to improve. And as we improve, we are learning about the biology of these cells. I have two other questions to consider. I don't know how I'm doing with time. And, you know, I like to bring this up, especially when I'm talking to my students. I think these are some things that are important to think about. So do organoids need to faithfully recapitulate the structures and functions of the in vivo organ? And how good is good enough, right? So like how well do they need to recapitulate every feature of an organ for me to be able to ask the question that I want to ask? And then how do we validate findings from organoid models? Again, we're trying to model human biology that maybe we're not able to model in an animal system. So how do we know that what we are seeing in an organoid is valid? And so I like to finish so that I can talk a little bit about this with some of the new NIH priorities that came up recently with this prioritization of human-based research technologies, which, again, pros and cons, and we can have a discussion about this for hours. But I think for this group, I think it's important to think that this is something that NIH is looking for. And not only that, but they also just launched this NIH Standardized Organoid Modeling Center that, you know, with the idea of generating a reproducible and standardized organoid-based method. And just with a note from Emory and from my own lab, also for this group, I think it's important. And this is mostly for brain, but we, about 2 years ago, together with Stephen Sloan and Fikri Birey, we launched this brain organoid hub at Emory in the Department of Human Genetics with the idea, again, of standardizing our cultures. We were very lucky to have 3 of us in one department working with organoid models. All 3 of us come also from the Pascal lab, so we knew each other, and we decided to kind of join forces and work together towards making a more reproducible model system, and standardize it. And so yeah, we basically have a goal not only of making organoids for our own labs, but also we do tests, and we try to make the organoid more reproducible. We have full-time staff that actually make organoids for the 3 labs. Then they do technical tests, and also a big part of it is data sharing and collaboration. We share all of our protocols online, and we help people to get started. We consult. And we have had a workshop in the past where we invited people come and learn about organoids. And more excitingly, recently we got this automated iPSC and organoid machine. It's called the Cell Express AI from Molecular Devices that basically can do everything. So you can put your iPSCs, it can culture iPSCs, aggregate, and make organoids and culture them over time. It has AI capabilities. It can take pictures over time and can tell you when your cells are ready. And it's not just for brain, it's for all types of organoids. We just got it installed in the hub. And so our goal is not just for us 3 labs to use it, but to be able to be a resource for the Georgia community looking to get started with organoids, and kind of lower that barrier for people to get started. And we're not there yet, so we still have a lot of testing to do with the machine and making sure that we can get it up to speed. But then, we're hoping that we can get there at some point soon. And if you're interested, reach out. And I think that's all I have for today. Yeah, thank you. And yeah, again, I'm very broad, but I'm happy to answer questions and kind of discuss some of these topics.
Thank you, Jimena. That was really helpful. There's a question in the chat already, and I was going to ask you also to if you would put your website in for the Organoid Hub so people know where to go when
Yes.
are interested. I realize you're not quite ready yet, but when you are. So do organoids have innate adaptive immunity if they're avascular?
Yeah, no, the answer is no. And again, this is mainly brain that I know about, but they don't, yeah.
Okay.
They have what an astroglia can have, basically. And I'm putting the link to the hub in there.
Candace, you got a question?
Yeah, Dr. Anderson, nice presentation. So I am fresh from the Cold Spring Harbor organoid course last week, because I'm trying to meet this organoid thing. And what I can mention from that course for 4 days of organoids there at Cold Spring Harbor was that most people are really using these types of bioreactors that have an endothelial layer on the bottom, and then they replicate this vascular flow. That seemed to really be state of the art across different platforms. Is that something that you are thinking about incorporating in your organoid hub, or is that kind of, where does that sit in your landscape?
Yeah, we're not. I'm not super familiar with what the state of the art in that is. I mean, I know of this kind of organ on chip where they have this endothelial and they put motor neurons mostly and other neurons in there. And yeah, again, it's the question of how good is good enough. So like while we do try to innovate and improve our organoids, we don't continuously try to change our system, right? Because we want to ask questions about specific, specific disease. And we started with our regular spinal cord organoid, and we kind of are using that, and we're happy with that to answer the question that we want to ask. But again, it's like if that's important for your question to have an endothelial component, then absolutely, I would go ahead and try to incorporate that. And I think that would be a little bit more hands-on that the suspension cultures that we currently grow. So like, using it for example, a robot, it would be a little bit more tricky, less standard, I guess. So yeah, again, it really depends on your question. And if endothelia are something that are important, then I would invest in getting it set up.
There's a couple questions in the chat. So any experience or future plans for skin-related organoids incorporating sensory neurons and immune cells to study neurocutaneous inflammation?
Not me. I don't know. I wonder if anyone in the group yeah, knows or has any interest on that.
If anyone does, please put your name in the chat and connect with Dr. Herrera. Another question, is there any commercial device or straightforward pipeline to vascularize organoids?
Not that I know, not straightforward. Like there are a couple of papers showing, and there's different, I guess, options. Like there is an assembloid where you can have a brain organoid, let's say, and then you can make like a vascular organoid and then assemble them together, and then the kind of endothelial will grow into the brain organoid. Transplantation is another way that people do, but all of these is like, I wouldn't say straightforward. Yeah, I think we're not in the like it's easy to do stage yet.
Another one is what's the difference between a spheroid and an organoid? I understand that spheroids are a 3D structure of a single cell line, but if you were able to mix multiple cell types to recapitulate the tumor microenvironment, would that be considered an organoid?
The term spheroid and organoid is, I again, started in the Pascal lab where we started calling them spheroids. But very quickly the field called them organoids, and we switched to calling them organoids. So for me, there isn't that much of a difference. I know that most people will think of a spheroid as a simpler organoid that maybe has like one or two cell types. And sometimes people will like make a spheroid of 2D neurons that kind of are aggregated together, and calls them spheroids to differentiate them from an organoid that is derived, by aggregating iPSCs and letting them mature into neurons. And so let me read your question again. If you were to mix multiple cell types to recapitulate the tumor microenvironment, would it be considered an organoid? I think so. I mean, I'm not very strict on what the name organoid means. I think it could be considered an organoid. There is a paper, and I can probably put it in the chat as well, about nomenclature in the organoid field. And I will look that up and I will paste it in the chat.
Okay. Maybe when we finish through the questions. So the next question is, are your organoids made of fully differentiated cells for studies in adult tissues?
Wait, sorry, I miss made of fully. Are your organoids made of fully differentiated cells for studies on adult tissues? I guess they're fully differentiated. They're not mature though. So I wouldn't consider them adult in any way. They're still embryonic / like a cortical organoid you can grow for about 250 days, and then you start getting a postnatal signature, but still very early on, not adult.
And they're not terminally differentiated, right? Like if you take them out of the differentiation media, they will go back to their stem-like qualities. Is that true? Or they stay at the that terminal differentiation?
Yeah, they will. Like unless you have, an organoid, you put, you start with iPSCs, you put your culture, your growth factors, and most cells will differentiate. Sometimes you get some progenitors that kind of remain there, but the cells that differentiate will remain differentiated. They won't go back if you remove. So like we put differentiation media for some, like a month maybe, and then after a month they are on just near basal media, very basic media. We don't put anything to keep them differentiated. Once they're differentiated, they stay.
Okay, next question. Would you know if there are gut organoids that can be used for microbiome studies?
I don't know. I would guess so, but I'm not familiar with it.
Yeah, tons, I think. Like I said, I just came from that organoid meeting, and maybe there was 15 or 20 talks on gut organoids and microbiome studies. So I think that's a pretty hot field.
Okay.
Yeah, we're doing a lot of that, Kathy. So who asked the question? I can, if somebody wants to ask that question again, they can email me directly. We're doing that work in my lab.
All right, so Kenneth, you should email Renat Jones, and we're going to get to everybody's work in just a minute, but thanks to everybody for chipping in. It's great. Another question, what's your opinion on incorporating 3D printed or bioprinted components to generate more complex tissues, including long-term perfusion?
Yeah, definitely an approach that people are using. My colleague, Stephen Sloan and Emery, is doing something like this to incorporate vasculature actually, and to pattern the organoids in a more controlled way, to introduce gradients in, that's my son, I don't know if you can hear him laughing, introduce gradients of morphogens in there. So again, I think I would love for a time when this is standard and easy to do for everyone. I think again, it depends on your question. If what you need is a more complex tissue, then go for it and try to incorporate it. But with what comes with it is that it just requires a lot of characterization. So like every iteration that you do of your organoid in terms of implementing something new, you kind of need to recharacterize it, right? So like you need to ask again, what cell types do I have? Do they mature? Are they functional? Right? So like if you're gonna now add endothelial cells or you're gonna add a matrix, you're gonna have to ask all these questions again. And so like it's depending whether you want to spend the time to characterize them again or you want to use a protocol that is already characterized and you know what you're getting.
Right, I think maybe the last question for this part is, do you have, or are you planning to implement in the Brain Organoid Hub organoids of distinct cortical areas?
Trying to think if we have. I don't think we have implemented that yet. I know some groups working on it, and it wouldn't be very hard to implement. My guess is that it's just adding a couple of extra factors to your recipe. So it would be simple to implement if that was of interest.
Okay, and then so this one's for the whole group. If there's anyone interested in working in tonsils-derived organoids, particularly to assess TFH development. So thank you, Jimmy. That was really eye-opening, I think, to help some level set the expectations of the field. So we're going to take some time now to hear from you so everybody can share what they're working on. Remember, what we'd like you to say, you have to do this in under 1 minute because we have a lot of people on the call today. So in under 1 minute, tell us your name, your institution, what you're working on, and what you're looking for in terms of a collaborator. I'm going to call on you by where I see you on my screen. And so starting with Candace.
Okay. Hi, I'm Candace Floyd. I'm at Emory University and Department of Emergency Medicine. My team is interested in understanding how occupational exposures such as infrasonics, blast exposure, things like that might affect organoids in a way similar to traumatic brain injury, to understand genetic susceptibility to long-term consequences from traumatic brain injury. So we're interested in multicellular brain organoids to use as a screening program for identifying GWAS identified susceptibility, TBI susceptibility genes.
Thanks. I think what we're going to see here is you'll find other people who are interested in developing organoids in your tissue. And those are the people you want to connect with as we go forward today. Reynald.
Hi there, yeah, Reynald Jones. I study host cell and microbe interactions in the gut, and I use gut enteroids to look at the influence of bacterial generated metabolites like short chain fatty acids, indoles, betaines on gut development and permeability.
Thanks. And you are from pediatrics at Emory.
That's correct.
Okay, Martin.
Hello, everybody. My name is Martin Tomov. I represent the New Emory Innovation Centers. So we have a range of tools and facilities that allow bioprinting, tissue engineering, biomaterials, and microfluidics integration to make more complete systems, kind of like these NAMs that are now all the rage. And we make these resources available for Emory and associated institutions to provide a hub and knowledge on how to integrate them so that research questions can be moved forward faster.
That's great. Thanks for that. It's a great resource. Martha.
Hello, everyone. My name is Marta. I am instructor in the Department of Orthopedics here at Emory. So I am working with iPS cells and MSCs to differentiate them in chondrogenesis and osteoblasts, all these kind of tissues. I'm trying to implement some like interaction between different cell types, but just I am, starting to learn about this.
Okay, great, thanks. Christian.
Hello, everyone. So I'm Christian. I'm in the Department of Pediatrics, Division of Neurology, and I'm our lab. We grow brain organoids. So, but now we are interested in studying the physical factors involving the brain and the brain cancer invasiveness into these organoids. So, if you are interested in talk about how cancer can be together with organoids, we are very interested.
Great, thanks. You followed the directions and you said what you were doing, you said what you're looking for, so that's great. Julia.
Hi, my name is Julie Babincy. I am in biomedical engineering at Georgia Tech, and I am interested in the immune system. I work with dendritic cells and culture them in biomaterial gels, and I'm interested in immune organoids. And I also have interest in multiple sclerosis and type 1 diabetes. So any kind of studies with that, I'd be interested with organoids too.
Thank you. Christopher Porter.
Hi there, I'm Chris Porter, Department of Pediatrics at Emory, and we study leukemia, B-cell acute lymphoblastic leukemia in my lab. We'd be interested in organoids that can model the immune microenvironment and ways to modulate those to treat the leukemias.
Hearing a lot of immune as we go forward. Bob.
Bob Taylor, cardiology. We are at Emory and we are studying sickle cell disease and changes in gut intestinal permeability. And so we're interested in connecting with folks that could help us with organoids in the intestine. And I've already connected with Renault by text here.
Awesome. That's what we want to see. Urellis.
Hi, everyone. My name is Jadalis Gonzalez Vargas. I am a postdoc at Georgia Tech and in the Georgia Clinical and Translational Science program, TL1. My work is in lymphatics. I do lymphatic spheroids and organoids. I've been working on this for about 6 years. I did my PhD on that. And right now, as a postdoc, I'm looking to go in more into genetics. So I study genetic mosaicism, specifically in lymphatic malformations. So I am looking to connect with more people that are more into the iPSC research to develop these into iPSCs into lymphatic endothelial cells, which I have some experience, but genetics are still a challenge.
Thank you. Yun Wang.
Hi, I'm Young. I'm a assistant professor at Emory's Biomedical Informatics Department. My research mostly focuses on leverage AI MRI to investigate early brain development. I'm interested in organelles, specifically in how investigate different medication affecting the placenta organelle and then potentially the brain organelle. So hopefully I can find a collaborator. I'm also starting doing a pilot using high field MRI to imaging the organelle. So yeah, just want to explore a little bit. Thank you.
Thanks. Charles? Charles Wang, are you with us? Okay, Corey?
How you guys doing? My name is Corey Wells. I'm a PhD candidate at Emory University in the cancer biology program. And due to the changes in the NIH, my lab is actually looking to develop pancreatic PDX organoids. We recently started working on this, so we're just looking for collaborators and or people that could help us work our way into the field.
Okay, thank you. Jürgen?
Good afternoon or good morning. I'm from Germany, the German Airspace Center in Cologne. And our special interest is to fly neurons in suborbital flights on sounding rockets, as well as on commercial space stations. And because maybe you have heard about Artemis 1, NASA, the surrounding the moon, we had a very nice radiation experiment, MARE, where we had human torsos and detected the radiation during a flight mission around the moon. Next year, there will be 4 astronauts flying around the moon and to prepare experiments for crew health and for countermeasure development. The work on organoids, special neurons and nerve cells would be very interesting to have them on suborbital flights as well as in future lower Earth orbit commercial space stations. So if you are interested to connect, you find me under my email, DLR German Airspace Center, Institute of Airspace Medicine in Cologne, Germany.
All right, I think you should talk to Bob Taylor probably.
Maybe Bob can connect. Do you distribute the emails afterwards?
We can do that. We can do that. We can create
participants. So I'm happy to connect with Bob.
Okay. You could also drop if you can drop your email in the chat.
Yes, I will. Thank you.
Sheik.
Hi, I'm Sheikh Rahman. I'm an assistant professor here at Emory School of Medicine. My lab's interest is immune reconstitution in HIV/AIDS, and we have been working on thymic organoids, artificial thymic organoids, regenerating T cells, understanding the mechanism of T cell regeneration, and also using this platform for understanding the HIV and simian immunodeficiency virus infection of the progenitors, as well as using iPSC-derived T cell generation. Also looking forward to have a more integrative form of organoids where we can also study, let's say, brain and immune axis using this organoid in combination with other organoids.
Okay, great, thanks. Tommy. Mizu, are you there?
Hello, can you hear me? Hey,
Yes.
hi, my name is Sami, pediatrics Emory. My work is next-gen sequencing in virology, including background is seek to understand viral host interactions. So I'm just here to learn. I just want to know how organoids is used to understand micro host interactions kind of thing.
Okay, thanks. Shulu? Sue, are you with us?
Okay, is that me? Shu Takayama here at, oops, let's see.
Okay, different one, but you
Different-
go right ahead. Go right ahead.
Okay, Shu Takayama, Georgia Tech. I'm a bioengineer. We work on organoid bioengineering. We do things like make inside-out organoids so that the apical site is out so that bacteria or virus can readily interact with lung epithelium or intestine epithelium, mammary, kidney. We also do some bone marrow. We also work on standardization of organoids like size, biomechanical properties. I'm also a co-chair of the Georgia Tech side task force to adapt to the new NIH initiative on new approach methodologies and things. And so hope to connect with many of you and learn and collaborate with Emory. Thank you.
That's great. Thank you. Sounds like a great resource too. Jairo Fonseca.
Hi everyone, I'm Jairo. I'm an instructor at the Department of Pediatrics. I am currently studying HIV latency establishments in 2D models of tonsil and intestinal tissue. And I have access to pediatric tissue to study on the ontogenic of the immune system, and that's my interest in moving to 3D organoids, because the way I would like to study pediatric tonsil 3D organoids to understand the immune responses against HIV during the formation of latency, and then test immunotherapies in these tissues. So I'm happy to be here and welcome any opportunity to collaborate.
Yeah, thanks. Sounds like you should connect with Dr. Rahman. Let's see. Dr. Pollack.
Yes. Hello, my name is Alexandra Pawlak. I'm from University of Georgia, College of Veterinary Medicine, Department of Physiology and Pharmacology. So I'm a vet, but I am working in the field of translational medicine. And I am working mostly with patient-derived cancer organoids. And I'm creating a model for co-culture of cancer cells with immune cells as a model for immunotherapy. Thank you.
That's great. There's definitely some overlap with what we've heard already. Paul?
Hi, I'm Paul Johnson. I'm the director of the Emory Primate Center. My interest in organoids is sort of twofold. One is that given the change in emphasis at the NIH, we're interested in a variety of different organoids that might complement in vivo work in non-human primates, and how non-human primate studies could validate or help refine those in vitro models. I also, as a AIDS immunologist, have a longstanding interest in T-cell development and have some experience in thymic organ cultures that dates back actually decades. So,
Okay, great, thank you.
if there are specific primate non-human primate applications or issues where access to non-human primate tissue for establishing organoids would be helpful, please contact me and I'll put my email in the chat.
Great, thank you. Lauren. Lauren, wait.
Hi, I am actually with SunExcel, which is a commercial collaboration of sites, and I'm just here to learn about organoids for the preclinical stage, because we work with a lot of pharmaceutical companies and biotechs that are doing very early phase. So being able to help recommend any sites within Atlanta that can use organoids and kind of help make those transitions and have a better understanding. Because I used to actually work at Emory on their phase 1 oncology team as one of the coordinators there. So, but now on the commercial side, but just again, kind of learning about and how we can use it early on with some of our biotech customers.
Okay, thank you. Maya? Lost Maya.
Hello. Yeah. Oh, can you
There
hear me?
you are. Okay.
Wonderful. I'm Maya Meeks. I'm with the Division of Animal Resources. I am a biosecurity officer here for our mice and rats and pigs on campus. And I'm just learning about how I can support the researchers when they want to use organoids in vivo. So I just want to learn about them. So when it comes up in your protocol reviews, I know what's happening.
Thank you.
Yeah.
We appreciate that.
Yeah.
Hunter?
Hi, I'm Hunter Jonas. I am an assistant professor in the Department of Pediatrics at Emory. I am working on developing an organoid model of the pediatric solid tumor neuroblastoma. The goal is to make a patient-derived immune-competent model so that we can use it to test the efficacy of adoptive cell therapies to translate to the clinic.
Sounds like you definitely need to talk to Hamina. Okay, let's see. Cecilia?
Hi, I'm Cecilia Botfield, a research scientist from Morehouse School of Medicine. My research interest is in iPSC-derived endothelial cells to model endothelial dysfunction in diseases like sickle cell disease. And so I'm looking for collaborators who are also probably doing similar work, and I'll be happy to learn from them.
Thank you. Wagbozo? Wagbozo, are you with us? Okay, does Ms. Pina?
Hello, everyone. Hope this is working. I'm Despina. I'm the Director of Research Projects for Dr. Deborah Brunner's lab at the School of Nursing, Emory University. And we are studying how the vaginal microbiome affects postmenopausal women that undergo cervical cancer therapy. And we have seen some strong associations with treatment outcomes, treatment-related toxicities, as well as cervical cancer recurrence, which happens in about 35% of this population, unfortunately. And we are looking for collaborators to learn how we can start testing some of the hypotheses that we have found in our studies, how those microbes affect specific outcomes. Probably we would like to start from simpler in vitro models, but here to learn and connect with people. Thank you so much.
Thank you. Melanie. Melanie Grant. Okay, Jenny.
Hi, can you hear me?
Yes.
Hi, I'm Jenny Shim. I'm in the Department of Pediatrics. I actually work with Hunter Jonas, who just talked a little bit ago, in the pediatric cancer neuroblastoma, which is actually not a brain tumor, but outside of the CNS. We're looking at patient-derived xenograft organoid models, cell line models, outside of the immune compartment of things, I'm interested more of the extracellular matrix and also the angiogenesis component of things. So if anyone wants to collaborate, that'd be awesome. Thank you.
Thank you. Luis?
Hello everyone. I'm Luis Arrieta, a postdoctoral researcher at Georgia Tech. Where I work is in the Andres Garcia's lab. My research focuses on synthetic biomaterial for organoid culture. Specifically, I'm developing PEG-based hydrogel to support the culture of patient-derived enteroids or intestinal organoids. I designed scaffolds to better mimic the native intestinal microenvironment. And additionally, I work in gut-on-a-chip devices that integrate both intestinal and immune components to model the inflammatory response and study the gut immune interactions in a more physiological, relevant way.
Yeah, I definitely heard some other people interested in gut here today. Let's see, Daniel. Oh, no, Daniel has not talked yet.
Oh, hello, everyone. Thank you. My name is Daniel. I'm the Associate Director for Research Projects at the Emory University Department of Dermatology. And our team is mainly working in studying inflammatory skin diseases, mainly hidradenitis suppurativa and bullous pemphigoid. And we are looking for a better human model to address neurocutaneous inflammation, especially the interaction between sensory neurons and immune cells. In relation to itch and pain, which are the main symptoms that affect patients. Thank you.
Okay, I want to direct everybody to the chat. Melanie cannot unmute for whatever reason, and so she put what she's interested in the chat, so please respond to her there. So I'll give the last couple of people who we didn't hear from a chance. Charles, Shu, or Wagbozo, are you here? Apparently not. All right, so thank you. So now we'd just like to take a few minutes here at the end to talk about what did you hear today that is interesting or burning, or what directions do you think that a collaboration among the people who are here could help move this field forward? I think you heard several people talk about resources. I mean, Jimena and Martin and Georgia Tech. I lost you, Georgia Tech, Shu from Georgia Tech. So those are great resources, but we are also interested in fostering collaborations between researchers with similar interests. So what did you hear today that was interesting or really sparked your enthusiasm? Don't believe you heard nothing.
Seems like there's a gut group that ought to get together.
Yeah, definitely gut group. And I heard a lot about HIV and immune system too, and some brain immune interactions. I know that I think Amanda would be interested in that.
I think brain immune and immune overall with different types of tissues. I think also, just to emphasize on the Georgia Tech side, we have a lot of biomaterials expertise. So if anyone's interested in that aspect, extracellular matrix or biomimetic materials, we have that expertise.
So generally at the end of a blue sky group, we ask the group what you would like to do to move things forward. And Renan, I saw you come on camera. Did you want to say anything before I close here?
Yeah, just it's apparent that there are a number of researchers, like what Bob said, on a specific working on the same type of organoids. So those guys can connect and consolidate resources, share expertise. That's what's apparent to me from this conversation.
And hopefully you'll be able to. So we have, in the past, we have put together a listserv. You can reach out to each other. We can provide email addresses to people who are here today, so you can connect with somebody who mentioned something that interested you. Lauren, the recording, can you make the recording available to people? As long as Zoom downloads properly.
Yes. Yeah.
Okay. All right. Is there anything you would like, anything that would be helpful to you beyond a potential listserv?
I have a comment. It seems that there's a lot of great research going on, but the understanding and knowledge of what resources are available to us as a group may not be necessarily well presented to everybody. So maybe if we can have like a small group, like the same type blue sky group where the resource people can kind of present in more detail what's available, what we can provide, and how that can be integrated in all of those cool projects we just heard about, that might help with kind of making sure that we all have access and understanding of what the resources and capabilities are present.
I think that's a great idea, Martin. And because it's such a great idea, I'm going to put you in charge of it. What we'll do is give you the emails of the people who talked about resources. That would be Jimena and Shu from Georgia Tech. I don't think he's on anymore. And you, anybody else want to be involved in that group? Takayama was the other one from Georgia Tech. So if you, we'll connect you with that, and if you could reach out to them and get that started, that would be terrific.
Yeah, sounds good.
And we can talk about how to advertise those resources. Jimena, any last words before we close?
Kathy, I'm sorry, can I just jump in real
go ahead.
quickly? Just the Emory Primate Center has an annual pilot program. One of the challenges that I think underlies all these discussions is how do you get something off the ground and get, put together the pilot data to get a system going. But we have a pilot program, and the development of new approach methodologies, NAMs, including organoids, will be one of the priority areas. That the pilot program RFA comes out in late November, early December, and generally our due date is around February, and it provides up to $70,000. So that may be an opportunity that people want to think of. Again, non-human primate tissue would need to be involved in some way, but organoids and NAMs are a particular focus of interest for the upcoming cycle.
Thank you, Paul. That's a great resource. If you would, when your call comes out, if you would send it to Lauren or I, we can send it out to this group.
Okay. Yeah, we will add that. We try to distribute as broadly as possible, but we'll make sure we hit you all as well.
Okay, great. Thank you.
Final note, Kathy, sorry, there's also a resource that everybody should be available. There is a core facility at Emory, and it's called, I wanted to get the proper name of it, so it's called the Emory Stem Cell and Organoid Core. So if you're looking for resources, there might be some expertise within that core that'll help you in your research. So if you Google Emory Stem Cell and Organoids Core, then you will, that's another research to tap into.
So thank you. That's great. So Martin, maybe you could include someone from that core in your small group. And also remind everybody that we have an agreement among the different institutions in Georgia to share cores. So you will, even if you don't work at Emory, you have access to that core facility. Anybody else want to comment before we close? I'm gonna have the last word.
No, I mean this is great, and hearing so many organoid people that are close by is very exciting. And yeah, looking forward to hearing more and working with, I forgot, was it Martin? No, yeah, to like generate some resource list.
So please take note of what's in the chat. There are multiple people who have posted things in the chat about the kind of researchers, collaborators they're looking for. We had the QR code at the beginning. If you do find someone you want to collaborate with and you need a little bit of money to get you started, reminder about the mini grants, and Ray just posted a link to that in the chat. So with that, I think we'll end for today. It's been a really interesting discussion. I think there's lots of opportunity here, and there's lots of reason why we all need to think about organoids because of the focus of the NIH. So thank you all for participating. We do have a small survey, if you would, that gives us feedback on how to make these events run better, be more useful to you. So if you take a minute to complete the survey, or Greg can send it out afterwards as well. And we really do value the feedback. So thank you for everything, and good luck.
Thank you.
Thanks to everybody. Thank you.
And Jimena, a special thanks to you for putting that the talk together to get us off the ground. That was terrific.
Thank you, Kathy. Bye.
Bye.
Thank you. Bye.
Bye.